for successful cell culture. However, even the average Joe can ace aseptic technique. See how Joe placed his water bottle outside of the lab in accordance with the lab's regulations posted outside of the door. Personal protective equipment, or PPE, protects you and your experiment from contamination. Be sure to wash your hands thoroughly before putting on gloves. 30 seconds or more should do the trick. Turning off the faucet with paper towel provides an extra level of protection from contamination. Good job, Joe! Select gloves that fit properly and make sure that the type of gloves used is appropriate for animal cell culture. And remember, your PPE is never complete without a smile.
Yay!
Turn on the biological hood. If the fans were not previously on, let them equilibrate for approximately 20 minutes or however long your specific hood recommends. Open the hood to the indicated height. Spray the work surface with 70% ethanol to disinfect. See how Joe is careful about selecting media and buffers that are his. This way he knows what is in it and if they are sterile or not. Joe will warm the media to 37 degrees Celsius before using them in the hood and with cells. This could take some time, so plan ahead. Notice that Joe keeps track of what he put in and only removes those items from the water bath. Before putting the bottles in the hood, Joe disinfects them with 70% ethanol. It is good practice to disinfect everything that goes into the hood, even stereological pipettes. Labeling tubes helps prevent confusion and accidental contamination. Notice how Joe's workspace is set up to limit how much he waves his arms over all the containers. This can vary for each individual. Also note how Joe is working in the middle of the hood and not over the grate. This keeps the air flowing and everything sterile. When unwrapping a stereological pipette, open from the end that attaches to the power pipette. This way you never have to touch the parts of the pipette that go into sterile containers. If the pipette touches anything outside of the sterile bottles, it must be immediately replaced. Always try to keep the cap on sterile containers, removing only when necessary, and never touch the inside of the cap. If you cannot properly hold the cap, simply place it on the hood and push it to the back out of the way. Right, Joe? Right. To keep cells in ideal conditions and away from contamination, make sure they are the last into the hood and the first out. Always check your cells to make sure they are healthy before using them. Beautiful, just beautiful. Even the bottom of cell flasks should be disinfected before entering the hood, but be careful not to spray ethanol into the cells. Once the experiment is complete, place the cap on the cells loosely to allow for gas exchange inside the incubator. Some caps are designed to do this while tightened, so be sure to know what kind of materials you're working with. The cells are the first thing that Joe puts away, before he puts away the rest of the bottles. Disinfect the hood again before leaving. Make sure you wipe up anything that might have spilled as well. If the vacuum was used for waste, spray ethanol into the hose before turning it off. Shut down the hood, but ask your instructor if to keep the fans on or to turn them off. Properly discard of all materials that may have touched biological waste. Never wear your PPE outside of the lab, even if it does look hella cool. Always wash your hands before leaving, just in case. Don't forget your water bottle. And that's how to master aseptic technique.
Now we will learn how to passage non-adherent cells. In this case, we're going to be using Jurkat cells. Always have a protocol ready to go so you know what you need to do and when. It's also helpful to check for all the reagents materials needed a day before you plan to do an experiment. Remember that all the rules we learned about aseptic technique in Part 1 still apply. Always check the cells to make sure that they are healthy. Note any oddities in their appearance and the color of the media. Using a stereological pipette, remove half a ml of cells. Be sure to swirl the cells before you remove them to make sure that they are evenly distributed. From that half a ml aliquot, take 100 microliters out and put it into a different microcentrifuge tube. Then accompanying this with another 100 microliters of trypan blue. This makes your 1-to-1 dilution. Always keep the cells in the flask inside of the incubator while you're counting the aliquot you set aside. To count the cells, you'll be using a hemocytometer and the microscope. Always clean the hemocytometer with ethanol before and after each use. From your 1-to-1 dilution, you'll remove 10 microliters and put it under the cover slip. This can take a few tries, so don't feel discouraged. Under the microscope, the hemocytometer is broken up into different quadrants. Count as many quadrants as you would like and how many cells are inside, but divide by the total number that you did. Alive cells will be white and dead cells will be blue. The percentage of alive cells is known as the differential. After you do the math of how the average count was times the dilution factor times 10 to the 4th, you can figure out how many cells you need to transfer to a new flask. If you're ever questioning your math, always ask a friend or the instructor for help. Then dilute the subset of cells with the amount of media needed from your calculations. Loosely put the cap back on and label your flask. You should include the cell type, the passage number, concentration, and date. Always remember to clean up your waste. You can use the vacuum to clean up the old cells from the old flask. And that's it. Simple as that.
These are like not stick stop. These are like.
Check with the timer. The timer has a magnet on the back.
No.
There's my waist. No. I need something for weights.
Should we include a picture of the people?
Joe's frustrated at my terrible lab partner, Taylor. She's not following proper aseptic technique, and it's ruining everything.
Joe. Joe.
Oh, I can get on this. Taylor! We need to clean this up. This is ridiculous. Can't work in these conditions. Taylor. Taylor. None of these are labeled.
Label them then.
Taylor, what are you doing? That's not how it works. Okay. That's okay. I'm done. Taylor, did you take the PBS?
Joe, look what I can do.
Good job, Taylor. Good job.
You just touched your face with your gloves on.
Let's take the gloves off. Okay, we can stop. Taylor, these numbers don't make any sense.
Yeah, that's why I just changed mine.